This is Project Neuroscience. Today, we're talking about the neuron. At the end of the video, there will be a list of all the new terms we discuss. The neuron is the basic functional unit of the nervous system. Like other cells of the body, the neuron contains a nucleus, an endoplasmic reticulum, mitochondria, and other membrane-bound organelles. The prototypical neuron contains dendrites, derived from the Greek for tree, which act similarly to an antenna, receiving signals from other neurons. The soma, or cell body, the axon, which is a long projection beginning at the axon hillock, proximal to the soma, and ends at the axon terminal. The axon is a lot like a fiber optic cable, which sends electrochemical impulses to other parts of the nervous system. It is surrounded by a fatty myelin sheath composed of an oligodendroglial cell, which acts as the insulator of our cable. Dendrites and the axon are collectively known as neurites. The axon terminal is the site of neurotransmitter release, our chemical messengers, that transfer information across the synapse, a gap between the axon and a neighboring dendrite. Neurotransmitters are contained in numerous synaptic vesicles that measure about 50 nanometers in diameter. We will talk about neurotransmitters in greater detail in an upcoming video. Because the axon does not contain ribosomes, all protein must be synthesized in the soma. To reach the axon terminal, protein must be transported down a molecular highway composed of microtubules and specialized transport proteins, collectively known as axoplasmic transport. Anterograde transport is the movement of material from the soma to the terminal. This movement is accomplished by kinesin, which walks material enclosed within vesicles down our microtubule highway. Retrograde transport is the movement of material from the terminal to the soma. This movement is accomplished by a different protein, dynein. Breakdown of this molecular highway is typically seen in individuals with Alzheimer's disease. There are many different types of neurons throughout the nervous system. As such, neuroscientists needed a mechanism to classify various types of neurons. Classification can be based on the number of neurites, types of dendrites, connection type or axon length, and by neurotransmitter. A neuron with a single neurite projection is unipolar, 2 is bipolar, and 3 or more is multipolar. Most neurons in the brain are multipolar. Next, a neuron can be classified by the shape of dendritic trees. Two broad classes are stellate cells, which are star-shaped, and pyramidal cells, which are pyramid-shaped. Pyramidal neurons are the primary signaling neurons in the cortical spinal tract, a tract between the brain and the spinal cord involved in movement. Classification based on connection can be divided into 3 main types. Primary sensory neurons, from sensory surfaces such as the skin and retina, motor neurons, which innervate muscle, and interneurons involved in neuron-to-neuron communication. Axon length is defined by either Golgi type 1 or type 2. Type 1 neurons are projection neurons with long axons. Therefore, type 2 neurons must have short axons and are involved in local circuits. Lastly, neurons can be classified by the neurotransmitter released. Acetylcholine, the primary motor neurotransmitter, are within cholinergic neurons. Notice that the suffix ergic is added. Gamma-aminobutyric acid, also known as GABA, the chief inhibitory neurotransmitter, are within GABAergic neurons. The same process can be applied to serotonergic neurons, etc. While much still remains unknown, glial cells serve as support cells in the nervous system. Astrocytes fill the space in between neurons and play a role in taking up, or reuptake, of neurotransmitters. Additionally, astrocytes maintain the extracellular concentration of key substances such as potassium, which will become important in later videos. Oligodendroglial cells wrap, or myelinate, axons. In the peripheral nervous system, oligodendroglial cells are called Schwann cells. Myelin aids in signal transduction by containing ions within the axon. Breaks in the myelin sheath are known as a node of Ranvier. These nodes allow the axon to exchange ions with the extracellular fluid. These nodes are very important in the propagation of the action potential. Lastly, a class of cells called microglia function as phagocytes to remove debris left by dead or degenerating neurons and glia. You can now pause the video here and review the terms. Next time we're talking about resting and action potentials. If you like what you saw and want to learn more about neuroscience, then hit subscribe for new videos every week.